I would like very much to introduce Dr. Eric Betzig, who is one of our Centenary speakers this year, and I could think of no one better to help us celebrate 100 years of health and medical research as an institute. Eric is currently a group leader at Janelia Research Campus in Virginia. He did his doctoral work at Cornell University and then worked as a PI, a lab head at the Bell Labs, AT&T's Bell Labs. And we were talking about the model that the Bell Labs created over many years, and having just seen the success rate of National Health and Medical Research Council funding plummet to less than 13%, there's something really desirable about an organisation that brings researchers in, gives them money, insists they don't apply for grants, and just allows them to go and research in a broad area, anything that takes their fancy. I think that's something that institutions can aspire to. And in my slightly libertarian moments, I wish that our institute had an endowment of $2 or $3 billion rather than the current endowment that it has, so that as I recruited laboratory heads and postdocs and students, I could have a clause in their contract which forbade them from spending time applying for grants. So if we have any donors in the Institute, in the auditorium, I'd be absolutely delighted to talk to you, especially if you're an iron ore magnate or one of the wealthiest women in the world. I'd be willing to change the name of the Institute. I digress. It was while at the Bell Labs that Eric began the development of super-resolution microscopy. He tired of that world and resigned, and in 1995, while unemployed, and I like this as well, published the concept that would be developed into that technology. He worked at a startup to try and develop that and resigned again in 2002 when there were some challenges commercialising. And then with his Bell Lab colleague, Harold Hess, he utilised photoactivated fluorescent proteins to really bring super-resolution microscopy to reality. He was awarded the 2014 Nobel Prize in Chemistry, after which he then published a paper on light sheet microscopy, which has really revolutionised the field again. And as I mentioned at the start, he's currently a faculty member at Janelia Farms. Eric, we thank you for taking the 28 hours to fly here. We hope you enjoy your walk in the Dandenongs yesterday, and we're absolutely delighted you can help us celebrate our centenary. Thank you.
Thank you, Doug. There's still seats up front, folks, so yeah, there you go. Don't be shy, there's lots of seats up here. All right. So anyway, thanks for the invitation to come here. It's always been on my bucket list to come to Australia, and this is my first time, and had a great hike yesterday. And congratulations on your first 100 years, and I hope it goes even better in the next 100 years. So I'm not a biologist, but it seems to me that one of the most interesting questions in all of science is how you go from inanimate molecules, which are produced stochastically and move stochastically inside of the cell, and then have these things self-assemble to create a cell which is not only animate, but robust under perturbation. And while there are lots of imaging tools one could use to try to unravel that mystery, including electron microscopy, by far probably the most useful tool conceptually would be fluorescence microscopy because of its protein-specific contrast and its ability to look at live cells. But there's always been this basic problem in that if that's the size of your protein molecule, then this fuzzy ball is the size of the spot you see from that protein molecule. And so it's too coarse of a tool by about an order of 2 orders of magnitude or a factor of 100. And so that's what's really motivated a lot of my career since the time I was at Bell, or even before that in graduate school at Cornell, going back to the early 80s. And so I worked on a number of technologies, which, as Doug said, failed one after the other. But the one that finally worked was one that I developed with my best friend from the Bell days, Harold Hess, where we realized that while those fuzzy spots are too big, if we can isolate the molecules into single fuzzy spots, we can find the center of the fuzzy spot to much better than the diameter. And the way you isolate it is by using something called photoactivation and certain fluorescent molecules that you can turn on with light. And if you turn on the light very low, then only a few come on and you can find their centers. And instead of getting this type of image, if they were all glowing at once, you get this image once you found the centers. So that idea, again, published while I was unemployed in '95. Harold and I were again unemployed in 2005 when we both flamed out as in industry and realized that we were really bad businessmen. And so we were fortunate enough that this was simple enough that we could do it in his living room. So we built the instrument there, and then were able to collaborate with some great people, Mike Davidson, an expert in fluorescent proteins, and Jennifer and George, who developed the first of these photoactivated fluorescent proteins and knew the biology that we as physicists did not. So we went from the idea of using photoactivation to all the data in our science paper that led to the prize in about 6 months. And it was because if you're really motivated, you can haul ass. And we were highly motivated as being unemployed. So here's some of the very first data of a cut through a cell, 80 nanometer thick section, a couple lysosomes. The single molecules are popping on one after the other, and as they turn off or bleach, others come on. And you start to build up that super resolution image, and you go from there to there to there to there to show the resolution. So with high enough labeling density, you can get down to about 20 nanometer resolution by this technique. And so 2005 was the luckiest year of my life because not only did Palm fall in our laps, but HHMI, which has, by the way, a $19 billion endowment now.
I was being modest in ambition.
They decided they needed a new money sink because you have to spend at least 5% of the endowment every year. So they decided to create their own freestanding research structure called Janelia. Harold and I liked the idea of spending somebody else's money instead of our own, so we signed up. And then for the first couple of years, we lived and breathed Palm. So here's some applications. Here we were looking at chemotaxis receptors in E. coli. We're able to show that the sizes and the distribution along the axis of these clusters were predictable in terms of a stochastic model of self-assembly, where the proteins are inserted randomly in the membrane and then stick with a probability to a cluster proportional to the size of the cluster. We were able to show that certain proteins, like in focal adhesions that are the points of contact to the extracellular matrix, that look co-localized at the diffraction limit are not, but form these nanoaggregates. We're able to show that certain core promoters are actually spatially segregated from the nuclear membrane where certain genes hang out. And this segregation is one way to silence the expression of those genes. And we were able to look in the dendrites of live hippocampal neurons and show that actin is polymerized only in certain discrete locations. So one thing about my history is that I get very excited about new technologies, and then after I sort of explore them for a bit, I get really upset and frustrated with them and leave them behind. And that's exactly what happened for me in Palm around 2008. So after playing with it for 3 years, I realized it had a lot of limitations. The first of these is that something called the Nyquist criterion. If you want to look at, say, a structure like that sine wave, if you had many molecules along it, it's easy to see, but if you had one only half period, you could miss it completely. That's known for any microscopy technique. You have to have enough pixels to see your image. What that means is you need very high densities of labeling, much higher than actually most pre-existing sample preparation protocols were doing. And so therefore it means that to do well with super resolution, this isn't true just of Palm, it's true of all the super resolution methods, a lot more falls on the responsibility of the biologist to be able to express and label at these densities than it falls on the actual instrumentation. And it's really, really hard. In fact, you have to do much better than that Nyquist criterion to get around the stochasticity of the labeling. That makes it even harder. The other problem is 95% of what we know about super resolution comes from looking at fixed cells. And it's known the purpose of chemical fixatives is to cross-link proteins. So you can see on this actin on a live cell and then fix, you lose a lot of the filaments. On the endoplasmic reticulum here, you can see all this aggregation that's happening under the fixation. So you can really ask, what the hell do we really know from super resolution on this stuff? And I would venture to say, unless it's a really robust stereotypical structure, not one hell of a lot. In fact, in my opinion, this Nobel Prize was really premature. It took them 53 years to give the Nobel Prize for electron microscopy, and they gave it for super resolution in 10 years. And there's still a hell of a lot of sorting out to do. Not that it isn't a viable tool and a useful tool, but we still haven't hit the kind of home runs that have been hit routinely with EM. So the other fork in the road, rather than looking at dead cells and fixed cells, is really what is the promise and the big potential of super resolution, is being able to see it at better resolution in live cells. And we had some modest successes with that. The first is a technique called SPT-PALM, where you photoactivate subsets of molecules and then watch their diffusion or assembly inside of cells. So it doesn't meet that Nyquist criterion of high resolution, but it has turned out to probably be the most significant and important biological tool in the super resolution toolkit in order to be able to study anomalous diffusion on the nanoscale, to be able to study aggregation of proteins and so forth. Eventually, we did demonstrate true live cell imaging by PALM at that sort of Nyquist limited criterion by looking at focal adhesions. But we looked at focal adhesions because A, they're super highly expressed, and B, those cells move really, really slowly. And so it would take a long time to get these images, like 25 seconds a frame, but that was okay in that particular case. So this is the broader problem in doing live cell imaging by super resolution, is that the two methods which shared the prize are something called STED, or a more recent variant called RESOLVE, developed by my co-laureate Stefan Hell, and then single molecule localization microscopy on this end. But if you just start looking at what's required in terms of the amount of photons that are needed, you need anywhere from 100 to 1,000 times as much light, or as many photons emitted, and the intensities are insane. Life evolved at 1/10 of a watt per square centimeter, and STED uses anywhere from 10 to the 5th to 10 to the 10th times as much as that. And PALM requires anywhere from 10,000 to 100,000 times as much as that. And then furthermore, because you're doing this point scanning instead, and you have to have so many pixels in your image, it's really slow. And localization, of course, is slow because you need to take thousands of single molecule frames to build up your super resolution image. So this brings up the broader point, is that there's always going to be trade-offs when you're doing any type of imaging. And so if you want higher and higher spatial resolution, it means you need to have more pixels in your image, which means you're taking more measurements, which is going to take more time, and it means you're throwing more potentially damaging light at the specimen. So if you're going to be honest, you're always playing one thing against another when you're doing imaging. And the history of super resolution is a bunch of egomaniacs like myself trying to push as far as possible up to that point to say, I got better resolution than you, and not really thinking about the consequences in terms of these other metrics. But there's a third method of super resolution that's useful for biology called structured illumination microscopy, where you put on a grading of illumination instead of uniformly illuminated, which allows you to demodulate information up to twice beyond the diffraction limit. Now, that factor of 2 limit of resolution is its Achilles heel, that it has a hard stop, whereas these other methods can go much farther. And that was the reason why the Nobel Committee said that they didn't include this in the prize as well. But that Achilles heel is actually the strength of structured illumination. Because first, you don't need such insane labeling densities to achieve that level of resolution. But second, it requires much lower intensities because you're not trying to get such resolution. And it's a lot faster, so it's much more live cell compatible. So the guy who understood these trade-offs earlier than any of us was Mats Gustafsson, who developed this technique first while a postdoc and then a PI at UCSF. And so here's showing some of the power of that technique, where we're looking at the endoplasmic reticulum Here for 1,800 time points. There is no other super resolution method that could look for probably an order of magnitude as many time points as this does without harming the cell or without totally bleaching the cell. And then here you're seeing imaging at 50 frames a second, the dynamics of the endoplasmic reticulum. And so no other method can touch these sorts of metrics. And you can see, yes, okay, it's only limited to 100 nanometer resolution. But there's a wealth of information in the dynamic aspect that you see in these images that you just can't get by any other method. And so we've been trying, so we actually were fortunate enough to hire Mats as a group leader at Janelia in 2008. And so he had the office right across from mine and was on a great trajectory. In 2009, he fell off his bike on the way to work, went to the doctor and was diagnosed with a glioblastoma and died of it in 2011. And so I ended up inheriting much of his group, and I've been trying to extend the legacy of SIM and to try to complete the story that I think he was working towards with this technology. So one aspect is, one of the Achilles heels of many super resolution methods is you're limited to thin samples because of out of focus fluorescence. But if you work with just grazing incidence illumination, you can just illuminate the bottom of a very large structure, like in this case, a Drosophila embryo, and still get good two-dimensional imaging. So here's an example where midway through embryogenesis, the epithelial cells come around. These are examples of the epithelium coming up in this purse string structure to enclose these amniocerosis cells from an earlier stage. These are these amniocerosis cells, and you can see now the single actin filaments going through these positive feedback oscillations of contraction and expansion that you can resolve with SIM, again, for very high speed, for very long number of frames. The other advantage of SIM is that you don't need specialized switchable labels like STED and localization microscopy require, so you can use any off-the-shelf fluorophores, in this case this VASP protein, which decorates the ends of the filopodia only on the epithelial cells and not on the amniocerosis cells. Here's another example of that multicolor imaging where we're looking at lysosomes trafficking along microtubules, and you can see different speeds of trafficking. You can see it actually switch between different microtubules, like different train tracks in this case. So we've had probably around 30 groups so far come to use our SIM microscope for live cell imaging at Janelia. And while oftentimes they start being interested in 3D imaging and so forth, in the end, many people gravitate towards doing things in total internal reflection. And so this, in TURF, you illuminate only the very bottom of the cell. And while it is restricted to studying in that region, you get major benefits because you don't do any harm to the cell above in terms of toxicity. You have a pool of molecules that can be recruited to the TURF zone, and you get very high signal noise because there's no background. So here's an example of that looking at a URCAT cell, sort of T-cell analog that's been plopped against a bilayer on a cover slip that has antigens that provoke the immunological synapse, and can watch the dynamics of that fast flow, internal flow of actin into that synapse here. Again, it only takes 1/10 of a second to get each frame. We did 1-second intervals here to match the speed of the process. So we've applied that in a number of ways with John Hammer's group at NIH. We looked at myosin 2. So it's been known that myosin 2 has 3 different isoforms, A, B, and C. And there was some debate about it. The field is that when myosins come together to form these large bipolar filaments, as to whether the different isoforms could intermingle or not. But by labeling the N and C termini with different fluorophores, we're able to show that all 3 isoforms could intermingle inside of these bipolar filaments. In a related experiment the following year, John's group was studying a little studied myosin known as myosin 18, and what we were able to show is that in vivo, myosin 18 actually makes bipolar filaments together with myosin 2, and that's shown here in this sort of 4-dot structure where the myosin 18 has the smaller chains and so are inside. And John's hypothesis is that the mixing of the myosin 18 actually changes the length of the bipolar filament, and hence the forces applied in actin-myosin contractions. and that the myosin 18s also form other types of anchorings to other types of targets inside of the cell. So if the knock against SIM has been that it's been limited to 100 nanometer resolution, and that's the reason that the Nobel Committee decided not to award it the Nobel Prize, then how can we go beyond that limit? Well, the simple and stupid thing is just to increase the numerical aperture of your lens. Olympus sells a lens that works at 1.7 NA in TIRF, and so we started to apply that, and with that we can push down to 84 nanometer resolution in the GFP channel. So here's an example of our focal adhesions like we did with Palm again, but now in 2 colors, and at speeds that are about an order of magnitude faster than we could do by Palm, and for many more time points. And so now we can then study the relationship of multiple proteins live as opposed to fixed, and show in this case that the paxillin has a sort of rearward flow, but the vinculin internalized at the center of the cell does not. If you like these movies, we had a paper that came out a couple months ago, and we put on a Vimeo album that has a lot of examples of this. Here's another example looking at these myosin actin contractions from the rearward side of the cell as the cell is moving on the cover slip. So we've applied that high NA SIM to study clathrin-mediated endocytosis. And because we do it in TIRF, the clathrin basket here is only illuminated at the very top, so now we then see a ring which is resolvable with the resolution we have with the SIM. And now we can then study from birth to death or internalization the whole lifetime of these clathrin-coated pits. And while you certainly could see the sizes of them by EM before, and you could see their lifetimes by optics before, never have you been able to correlate the two and actually look at the relationships between the size of the pits and how long they last. Another thing is, again, with the multicolor imaging, is we could then study the interaction of the cortical actin cytoskeleton with the clathrin-coated pits. And there's been a lot of debate in the field about the role of actin in clathrin-mediated endocytosis. While we haven't studied that exhaustively by any means, in the limited data we have, what we've seen is that actin is recruited right before endocytosis about half the time. But when it is recruited, there's a small but statistically significant shortening of the lifetime. We've also saw that the clathrin plaques, which normally look like big blobs in a regular microscope, are just basically aggregation of pits, and that individual pits spin out of these. We also found quite commonly a bunch of actin rings of roughly the same size as the clathrin rings. We originally thought that these would be involved in constricting the clathrin pits. Turns out that none of them are co-localized with clathrin. They're not really described very much in EM literature, but they're very common in the live SIM images, and we're really not sure what their role is. So with that high NA objective, we can get to 80 nanometer resolution, but what if we want to go further? Well, then we have to start doing the photo switching tricks similar to what Stead and Palm use. And so this was something that Mats was working on when he died with his grad student Hesper, and the approach they use is similar to what's done in what's known as Resolve by Stefan Hell, where you photoactivate all of your molecules in the sample, and then you come back and deactivate all of the molecules except those in the nodes of the standing wave of deactivating light. So this leaves these little spikes of remaining molecules which have high spatial frequencies to extend the resolution in Hesper's case down to about 50 nanometers. The problem with Hesper's experiment was on a fixed cell because it took 900 seconds to get one image. So that's obviously not going to be so great for live. So my postdoc Dong, thanks to a new fluorescent protein developed by our collaborator Pingyong Zhu at Chinese Academy of Sciences, came up with an alternative approach. And we realized that this depletion principle of turning on molecules and then turning them all off except for a few, which is the basis of the nonlinear SIM that Mats did, and also the basis of what Stefan does with Stead and Resolve, is really not a good way to do live cell imaging, because every time you transition the molecules, there's a chance you're going to lose them to some phototoxic event. And furthermore, it takes a lot of time and a lot of light to bring them up and then bring them right back down. They're not contributing any useful signals, the one that cycles, it's the ones that remain on that contribute the signal. So it's really photon inefficient and requires a lot of time and a lot of power, neither of which a cell is going to want. And so we came up with this alternative approach with Ping-Yong's protein, where we use a standing wave of light to not turn on all the molecules, but just turn on a subset, and then read those out with another standing wave of the same period. By doing that now with 2 standing waves instead of 1, instead of getting twice beyond the diffraction limit, you go to 3 times. So that takes you now down to 62 nanometer resolution, which is, if you followed this movie along, is pretty goddamn good, and actually becomes very competitive with any other method of super resolution, even on fixed cells, comparing them to the live cells, and there's no other method that can study live cells as well as this method does. And so this is an example of looking at the remodeling of actin over time with that method. We've used that also to study caveolin. So caveolae are another means of endocytosis inside of the cell. In the EM images, caveolae have a very stereotypical size of about 70 nanometers. But in our case, we find all sorts of ring-like structures of a variety of sizes. Plenty at that 70 nanometer size, but others that are larger. Some of these may be multiple caveoli docked around a vesicle, but it also highlights that, you know, with fluorescence, you're able to know exactly where all the protein is. With EM, you're always trying to infer. And if you don't have a priori knowledge, you may look at the things you want to look at and not see the bigger picture around it. So we only have one good photoswitchable probe to do the nonlinear SIM, but we can combine it with the linear SIM to do multicolor imaging. So here in the linear channel, we're looking at Rab5A as a marker of early endosomes, and then we're looking at actin in the nonlinear channel. And what we find is well, first looking at the endosomes themselves, is I picked 3 particular endosomes here to show that instead of seeing sort of a round shape, they show these very irregular shapes. And I chose those 3 because they look very similar to a 20-year-old EM paper I was able to find, so I was able to cherry pick to try to show that they look similar. And we see these dark holes that are probably either cargos or vacuoles. And we also see all sorts of different types of dynamics of endosomes, anywhere from random motion to corralled motion by the actin cytoskeleton to very fast active transport, probably along microtubules. So the moral of the story was that while we were all pushing to this spatial resolution at all costs, Mats marched in the opposite direction from the rest of us. And by doing so, while he gave up some here, he developed a much bigger application space with SIM by limiting himself to that 100 nanometer resolution. And so I tried to take that lesson to heart. And so in 2008, when I was as sick of Palm as I was of my earlier technologies in the mid-90s and would otherwise be ready to quit, I got smart because I was older. Instead of quitting, I said, well, let's see if I can do something else while I'm at Janelia. And so I wanted to ask myself, what if we backed all the way to the diffraction limit? Is there something we could do to make a microscope that would allow us to be able to look at the dynamics of living cells in 3D that would be as transformative in terms of how little the toxicity would be or how fast we could image by those metrics as Palm was transformative in terms of the gains of spatial resolution. And so the good news is that the standard tools people use for 3D live imaging leave a lot of room for improvement. They're basically the confocal microscope and the wide field microscope. The problems with these methods is that the high resolution information only comes from the single plane that's in focus, but both methods illuminate the entire thickness of the object in order to get their data. So you have all sorts of out of focus excitation that creates photo bleaching and toxicity. And so in my opinion, one of the most important innovations of microscopy in the past decade was when in 2004, Ernst Stelzer at the EMBL resurrected a 100-year-old idea called plane illumination. So here you don't bring the light through the same objective you detect with, you use a second lens, for example, a cylindrical lens, from the side to create a sheet of light which illuminates only the region near the focus. And by doing that, the other areas are dark, so there's no toxicity or bleaching there. There's no background to the image from those regions. You illuminate the whole plane at once, so you can use a fast camera and get your whole plane of data, pop, and then boop, 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 go up plane by plane through the specimen, get your image. So this has been transformative for studying embryogenesis at single cell resolution. But it has a limitation of its own, is that the laws of physics dictate a trade-off between how flat that light sheet can be over a given distance and how thick the light sheet can be. And over the dimensions of a single cell, it's going to be about half the thickness of the entire cultured cell, so you don't gain much benefit from light sheet in that case. So when I was sick of palm, this afforded an opportunity to see, can we make a thinner light sheet that would allow us to look at subcellular imaging inside of single cells? And the answer was pretty obvious, that first we started with something called a Bessel beam, which allows you to decouple the length of a little pencil of light from its cross-section. And by then eventually generalizing that on the basis of something called optical lattices, in a theory that I developed while I was unemployed before. Well, the history is that after I left my dad's company and was trying to figure out what to do next, I learned about GFP in 2003 for the first time. So I was the last man on earth to learn about GFP. But I knew that I needed to do microscopy again, and so I came up with this idea of using these optical lattices, which are used by the atomic physicists to trap atoms near absolute zero. And I figured I could make a multifocal excitation field for that. And eventually that evolved into this optical lattice microscope idea, and I was pushing that when Harold and I learned about photoactivation. We dropped the lattice like a hot potato, went and did the palm, and then that was that. But then eventually with the Bessel technique, we realized that instead of using one Bessel beam, we put in several Bessel beams and realized that that was even less toxic than having one. And so what I realized in 5 years of doing live imaging is that while the total dose of light that you throw on a live cell is important, by far a more important metric is the instantaneous power that you throw at the cell. And so what that means is that the confocal microscope may be the absolute stupidest possible way to do live cell imaging. Because not only do you have these cones of light above and below the focus that are doing all this damage, at the focus itself you have this actinic spot of light that's going, and it's leaving death and destruction in its wake. And so the moral of the story is you have to spread out the light. It turned out that the two-dimensional optical lattices are the ultimate expression of a multi-parallel Bessel beam array. And so I was able to dust off all of that old lattice theory that I had thrown in the wastebasket 10 years earlier and apply that to create these thin light sheets that were great for doing subcellular imaging, and that's what we call the lattice light sheet microscope. So here are some examples that were in our paper, and again, if you like these, we have a Vimeo album you'll see in a minute. Here's an example with Dyke Mullen's group at looking at cell motility in 3D through a collagen mesh, in this case looking at an HL60 neutrophilic type cell doing its dance, and you can actually watch as it squeezes through different parts of the collagen, expressing actin to create sort of a support structure as it goes through. We looked at dictystelia a lot. They're a light-sensitive slime mold amoeboid. And you can see as this movie cycles around, you'll see, boom, that lightning bolt. Our collaborators look at that and we go, what the fuck was that? And we go, I don't know, you're the biologist, you tell us, we don't know. But we've had like 50 different groups come to use this microscope. And every one of them goes away with the same feeling. I mean, it was great to win the Nobel Prize for Palm, but I am morally convinced that this microscope will be the high water mark of my career because everybody gets data that they love with this microscope. The downside is everybody comes for a week or two and they leave with 10 terabytes, and then they don't know what the hell to do next, and we never hear from them again because they're too embarrassed to ask us, right? We don't know what you can do with your 10 terabytes. But so the data analysis problem becomes huge. Our personal server has 300 terabytes on it right now from this, and that's all compressed, and we don't even store raw data anymore because it's easier to retake the data than to do that. So again, it's really fast enough that it's also easy to do multiple colors to get now the fifth dimension here. So here's an example where we're looking at histones to mark chromosomes, mitochondria, and the ER. And then we're taking this field of dividing cells and splitting it up computationally in 2-micron thick slabs so you can see what's happening inside. And you can see that the ER, instead of being that reticular network, forms these pockets. The mitos, instead of being sausages, fragment They fit inside of those pockets and they're carried along as the cell divides. Now note, this is 300 volumes through this field of cells at 3.8 second intervals. So in each of 3 colors, that's a total of 900 volumes. Each volume has several hundred planes. It's 250,000 2D images that are represented in this data. And yet, this is, cells are pretty sensitive during mitosis. There's all sorts of mitotic checkpoints where they'll shut down if they're unhappy. But all of these cells divide fine, and then their progenitors divide fine as well. So it's really off the charts compared to, say, a spinning disk in terms of both speed and toxicity, in terms of what you can get with this scope. So like I said, most of our collaborators are from 2 years ago are still figuring out how to chew on that data. So the papers are just starting to trickle out. So I'll give a few examples. So our collaborator, Yuko Momori Kiyosui at CDB Riken, was interested in EB1, a protein which is on the end of growing microtubules. So this is some of the raw data here. And then this is the tracking where they were looking at this EB1 that's on the end of the plus ends of the microtubules. You can see at different stages of mitosis that during prometaphase and metaphase, the microtubules actually grow faster. They grow fastest right near the centrosomes. And then if we look at that data further and break it down based on stage of mitosis, what you start to see is more and more tracks beginning to initiate near the metaphase plate instead of near the centrosomes as you go into anaphase. And so there's a question, are these just microtubules that stopped, went back, and then now are growing again, or are these new microtubules? So what they did is they looked at the trajectory of these new microtubules that were near the metaphase plate, and if they were coming from the centrosomes, they would have a predicted trajectory, but many of them form much greater kinks that would be unphysical if they were starting from the centrosomes. So that means that these are likely de novo nucleated microtubules right near the metaphase plate, which supports this Augment protein model of doing nucleation of microtubules during metaphase and anaphase at the metaphase plate. So here's another example that came out earlier this year with Alex Ritter, Jennifer Lippincott-Schwartz, and Gillian Griffiths at Cambridge, looking at, again, the immunological synapse, but now not artificially on a cover slip, but with T cells, primary T cells against dendritic cells. And so this is showing several stages of that process. Here's breaking it down here. And what they found is that prior to the granule release, there's this clearing out of actin, sort of ends up looking like a lamprey on the front end when the centrosome comes up and the granules are released. There's also this incredibly fast flow of actin along the sides of the T cell when it forms a stable, mature synapse in this case. So here's an example with Tommy Kierhausen's group who studies clathrin at Harvard Med. We're looking at 3 different planes sideways through this particular cell here. And what we can see is that right at the very leading edge, on the ventral side, the clathrin pits are quite stable and everywhere. But on the dorsal side, there are none at the very leading edge, but then at the junction between the lamellipodium and the lamellum behind it, you start to see lots of pits. And so what Tommy hypothesizes is what this does is it creates an imbalance. So the exocytosis is still going on everywhere, but the endocytosis is endocytosis is shut down at the leading edge so that there's more membrane therefore deposited, so you can get these sort of membrane ruffles to form that you see there. So in another example, this is now looking in C. elegans embryos with Geraldine Seydoux's group at Hopkins. They're interested in this protein MEG, which is responsible for granule assembly and disassembly of these P granules, these sort of aggregates of protein and RNA. And by looking in these 2 color channels here, by doing an analysis, we were able to show that the MEG protein is actually larger than this PGL protein, and that therefore actually surrounds the granule. But we also saw these sort of fluctuations in brightness, which were clearly at our signal noise, not due to just noise, but represent some sort of structure going on. So we switched, the other nice thing about the lattice light sheet is while you can sweep it along and get diffraction limited resolution, if instead you step it in discrete positions, because it's a structured grating, we can use Motz's principles of structured illumination to actually improve the resolution in that direction, in the vertical direction. And then what we saw is this much greater level of structure inside of these granules for this MEG protein. So it forms almost like a ribbon-like scaffold, which is highly dynamic through these P granules over time. So at the largest scale, we've looked at zebrafish, but in this case, looking just right at the nose and the olfactory epithelium with Marion Bronner's group at Caltech. This is just showing at one time point again. The lattice light sheet, while diffraction limited, because the light sheet is so thin, its axial resolution is twofold, at least maybe threefold better than confocal. And in shallow areas, it's much more aberration tolerant than confocal. So you can see the improvement, even though both of these microscopes are diffraction limited. Here's an example looking over many time points at neural crest stem cells, which are generated far away, and then they start to migrate, and as they differentiate, they start to express this green protein, and you'll see some of these guys express more and more green as they migrate towards this olfactory epithelium as they come in. So that's on the big scale. So what it's turned out is lattice has also proved very helpful on the very small scale. Because again, because that light sheet is so thin and is thinner than the depth of focus of the detection objective, if you park the light sheet at one plane in a thick specimen, you get almost turf-like signal to noise to be able to study single molecules. Now, in this case, you're looking at a 35 micron diameter spheroid of mouse embryonic stem cells. And if you use the standard wide field approach that people do to do single molecule imaging, the out of focus fluorescence just kills you. But if you can use the lattice light sheet, then you get great SNR. And this has now become a go-to tool in our transcription imaging consortium at Janelia. So in one experiment, what we do is we were looking at these SOX2 transcription factors and then studying the kinetics of their motion and finding that there's a two-component exponential to their residency times at different points, which they've been able to determine is due to specific or non-specific binding to the DNA. James Liu in the TIC was able to use this to then map out regions of hot transcription inside of the nucleus and show that they're spatially segregated from the heterochromatin in the nucleus. And in fact, next week we're actually giving up our lattice light sheet to James in his group and moving it up to his lab. So I swore to God I would never do single molecule localization microscopy again after 2008. In fact, I made a $1,000 bet to that effect with some buddies, and I lost that $1,000 because of this, because ultimately we realized that it was just too good of an opportunity to use the lattice to do localization microscopy. So remember I said two problems with doing single molecule localization. One, you can't look at thick specimens because of the out of focus. And the other was that damn Nyquist criterion, that it's difficult to get enough fluorescent label in there to decorate the structure densely enough at extremely high resolution to see it. Well, in the same year that we published the Palm paper, Robin Hochstrasser's group at Penn published a related idea, which doesn't use photoactivation, but uses the idea that in your media, you put fluorescent molecules that are just whizzing around on a Brownian motion. But when they stick and bind to their targets, instead of looking like a fuzz, then they look like these little bright stars and so forth. Then you can localize those. The advantage of paint is that you have an infinite army of molecules in that bath, and if you're just patient enough, you're going to get things sticking again and again and again, and you get amazing densities of localization. The disadvantage of the paint is that the entire media is glowing and your SNR is nearly zero. But with lattice, that solves that problem. So lattice plus paint is a marriage made in heaven. This has been used in thin samples in a microfluidic chamber to do very high resolution imaging, in this case, DNA origami, to look at these structures, and you can see, these are things that are just a few tens of nanometers big. But in that case, you're only limited to DNA. Here we were looking at an intracellular membrane marker that would stain all the membranes. This is the lattice light sheet image to begin with, and now this is the lattice paint image. as we go along. And so now, if you stared at enough localization microscopy images like I have, you get incredibly frustrated at seeing fields of dots over and over again. This does not look anymore like fields of dots. It looks like 3D EM, which is what localization microscopy should look like. So this is 500 million molecules inside of this one cell that were localized to see, all of these structures. So basically, what this also means is that we can then combine the lattice with the paint. And there's been a lot of work trying to do correlative electron microscopy to get the global contrast EM provides with the protein-specific contrast super resolution provides. But now we can get that global contrast by paint, so we don't need to go to a separate microscope and have these competing sample prep protocols between what EM requires and what fluorescence requires. And so this is an example where we were looking at the lamins for the nuclear membrane by palm, but then looking at paint. And here you can see the mitochondrial DNA in the Hooks channel that's looking at that, and it matches up exactly where there are voids inside of the mitos in the membrane channel. And the other advantage is that, again, you're not limited to thin samples. So here are two mitotic cells, one in prometaphase, one in metaphase, the Hooks channel to look at the DNA, and then the ZEP channel to look at all of the membranes. And you can see in this metaphase cell, in order to conserve surface area, there's all these little spiky projections off of the side of the cell here. And then again, there's also a scattering of DNA from the mitoDNA across the prometaphase cell that you see there. And at the largest scale, we've also even used this to look at multicellular specimens. So this is a fixed zebrafish, and looking at one of the neuromasts along the lateral line, the sensing organ. And so this is looking at a whole field of these cells here. In this case, this was 1 billion localizations. It took 10 days in the microscope continuously to get that data. But in the end, we're able to register everything to about 10 nanometer relative precisions to one another. So that relates to where I'm trying to go in the future, both with the lattice light sheet and with the paint localization, is, in my opinion, we've never seen the cell the way it truly is with any microscope. Because between the stress that we introduce with fluorescent labels, combined with the stress we do by looking at them in our standard microscopes, we're already looking at a highly artificial system. And then oftentimes we're looking at immortalized cells on cover slips, but that's not where they evolved. And so I think that we're on the cusp of possibly a revolution in cell biology to see the cell on its own terms. Lattice light sheet is part of the answer to that, to greatly reduce the photo damage inside of the cells. CRISPR-Cas9 is another part of that, to be able to express at endogenous levels. But the third part of the problem is we have to start looking at cells inside of the organisms where they evolved. And the challenge with that is that if you're going to use visible light to do that, is that particularly with the light sheet, is the light sheet is scrambled as it goes through. This is meiotic chromosomes in an adult C. elegans, and you can see we're fine here, and the light sheet gets scrambled by the time you go there. Here's a C. elegans embryo. On the detection side, if you only need to go through space in order to get to the objective, that's fine, but if your light rays have to traverse the embryo, they're scrambled before they get there. And so the light is scrambled in the same way that water on your windshield scrambles the light from the car headlights in front of you. The good news is that this was a problem astronomers had to confront half a century ago. And the solutions they came up with are known as adaptive optics. So of course, the light from the atmosphere, twinkle, twinkle, little star, will also screw up what's coming from their distant objects. So the way they deal with that is they shine a laser up in the stratosphere to excite fluorescence from sodium atoms to make a bright artificial star. And then the light comes back into the telescope, and it's now all aberrated by the atmosphere that's from the stratosphere on down. That aberration is actually measured with a device called a Shack-Hartman wavefront sensor. And then you can use a computer to put on a mirror that can change its shape to exactly balance the sort of warping that the atmosphere has provided to then get from a blurry image back to the diffraction limited image. Right now, if you don't use adaptive optics, the best, biggest telescope in the world won't do any better in terms of its angular resolution than a reflector you can have in your backyard. It'll collect a lot more photons, but the actual resolution won't be any better. But with adaptive optics, the best ground-based telescope kicked the Hubble in the ass. And the next generation of scopes, like the 30-meter scope and the extremely large scope that the EU is putting together, will just blow people away. I mean, the Hubble will look like nothing compared to it when that comes on with the AO that they'll have. And so, so we wanted, you know, I give a talk about once a year about the historical connections between astronomy and microscopy. And the take-home message of that talk is that the microscopists are the retarded stepchildren of astronomers. And that we steal everything from them about 50 years after they do that. And that's exactly where we are with AO right now. So, so a couple years ago, a group in Spain came up with a clever idea of just using 2-photon microscopy, which doesn't create a cone of fluorescence, but just a spot of fluorescence, to be able to create a guide star. We took that idea and added some other tricks of our own to that to be able to make a robust adaptive optic system, primarily for zebrafish. I'm happy using zebrafish as a model organism. I love the zebrafish. So this is an example where we're looking at a dense membrane label of neurons in the spinal cord region of a fish 72 hours post-fertilization. You can see that because the light rays are not coming to a common focus, the signal goes way down because you're not focusing correctly, but you also lose resolution and you turn on the adaptive optics and poof. So if you look on the right here, we're looking at a soma of a neuron 150 microns deep. Those are the mitochondria in purple. This is the membrane around it, but now we're going to turn on the adaptive optics and you go to there and you start to see the mitos. And then if we know the shape of our focus, we can do a deconvolution, and now we see the mitos as good as if this were a cell on a cover slip. And now you can see the membranes in the second channel. This is using a 2-photon mode to go deeper in the fish. This is now in the midbrain region, about 250 microns deep. If we turn off the AO, that's what you would see with a state-of-the-art 2-photon microscope. But now you turn on the AO, and now you get back to the diffraction limit. It's not super resolution, that's just diffraction limited. So part of what my job is right now is to educate the biologists who are doing developmental or neurobiology or any type of multicellular imaging and saying, if you think you're looking at the diffraction limit or whatever the NA of your objective is, I'm here to disabuse you of that notion, and that it's the deeper you go, the worse you get in a hurry. And so biologists don't yet really comprehend that AO is going to be the next big thing and is absolutely required to get to this ultimate goal of, again, being able to see the cell on its own terms and to be able to see those cells in the organism just as well as we see them when they're on a cover slip. So one of the problems with the method I just described is you need to be able to see that guide star. And unfortunately, a lot of biological samples have tremendous scattering. Just so it would be like trying to see the astronomical guide star behind a cloud. But one of the things that with my wife we've been able to do is that if you co-inject or co-express an infrared fluorescent protein, then you can see these guide stars at much greater depth, in this case throughout the whole mouse cortex, so we can get spine resolution, you know, down to 600 microns deep in the cortex, and then we get this great gain of signal in terms of doing functional imaging of neural activity. She's using that in her work to actually study the genesis of directional selectivity in the visual cortex. So the goal, you know, we have our Lattice light sheet, we have our AO. What we need to do is we need to integrate those technologies together. So this is some probably 3-week-old data now from the first experiments of going in the zebrafish at a membrane marker. And you can see this is without the adaptive optics. Now we turn on the detection correction to correct that arm. We had a separate arm of adaptive optics through the excitation, and then you add the deconvolution. Now you can see all the extracellular spaces between the membranes as you go down through the fish. One of the challenges in adaptive optics, which we've solved with the zebrafish, is that you can imagine as you go through different parts of the fish, the light rays are going through different material, and so you need different adaptive optic corrections. In this case, we're using 7 different tiled regions, and that big thing I showed you earlier, that was 20,000 different adaptive optic corrections. but it requires far less light or time than the actual imaging. And so our goal now, one of the things I would like to be able to do is get resolution like you saw in that one big image, but actually see that over time and watch the development of all the neurites and all of the wiring of the zebrafish over time. And I think we're on the cusp of having the technology in order to be able to do that. This is another example overlooking a larger field with a few more tiled regions as we go deep. But again, this is just the start of where we're going to get. We have a little slicking up still to do in this experiment, but I think this is going to be hopefully the next big thing. And so I'm pretty close to done. I'd like to say a couple more things. The first is about technology transfer. I mean, it's fine to win a Nobel Prize, but it really doesn't mean jack shit if it doesn't lead to biological discovery. And my feeling is that super resolution has had a few hits, but they've been more like singles and doubles rather than triples and home runs. And the only way they're going to, any of these techniques is going to have a big impact is to get them out of the hands of physicists and into the hands of biologists. And that's a difficult process. I mean, basically you have to cross this valley of death from something that I can operate to something a biologist can operate. And so we're taking a three-prong, but you guys know what to do with it and I don't. So, but anyway, so the three-prong approach. The first is for people to find out if they're interested in using these technologies. It would make sense to try before you buy. So with the Gordon Moore Foundation, we partnered to create an advanced imaging center at Janelia, where we have 5 of our in-house microscopes, the Lattice scope, the live cell SIM, Harold's exquisitely sensitive 3D eye-palm microscope, and Motz's multifocal microscope, which is great for 3D single molecule tracking. And so it's basically a 2-page scientific proposal plus your CV and biosketch. And if you're approved, you come for a week or 2 at a time. We pay for all your meals and on-site housing, and everybody can get drunk in the pub together at night, and then get up at 4 in the morning and take more data. So, and that's the typical way it runs there. And so the ASC so far is up to about 50 groups. My group alone had 50 groups with the Lattice. We've had about 20 with the SIMs so far. So we're really keen to have people come and find out if they're interested in these scopes. If they are, then the next stage is, first off, anybody who has the technical chops to build one of these themselves, we provide complete set of documentation. And so we've had about 50 groups execute research licenses for that. And then finally, it has to get commercialized. I couldn't care less about the financial money side of it, but I, having worked in business myself, I understand the importance of the D in R&D, and then what we do is this in a pyramid that's like this. And there has to be a lot of work to make something turnkey for a biologist. And so Lattice is currently in that stage. There's clones of our instrument available now from a company called 3i, and in a couple of years, Zeiss is going to have kind of a sort of a redesign from the ground up turnkey instrument that they'll offer. I'd like to see SIM follow that path soon. There's plenty of commercial SIMs out there, and one of my major frustrations in my life is that SIM has not been more widely adopted by biologists than it has, because I think it's the super resolution technique that has the most potential. And part of that is that the commercial implementations really have not been good. So I'd like to see that situation changes, and honestly, it's not that difficult to put one of these together. Certainly simpler than a Lattice scope. But in the end, getting this stuff out there is really important to me. And then, again, this is just to conclude. I have many, many people to thank, but I'd like to make 2 points in giving my thanks. First is that this was my group this summer. That was as big as it ever was. My group, everything I showed you in my talk from Palm onwards of the last decade was done by an average of 3 people. And I think that's our strength, to have a really small group. That was the strength of Bell Labs. That's the strength of Janelia, is that you're not vertically integrated, that you have to collaborate among groups, and you have to pull in outside people. And I honestly believe we're more productive with 3 than some Max Planck groups with 50. And I really think we almost work at an unfair competitive advantage with the small group that we have. And another part, the other secret weapon other than our small group size is the visitor program at Janelia. Is that Janelia is largely a systems neuroscience place with a sprinkling of imaging people or chemists and other things. But the reason we've been able to be so successful is because it's been so easy to bring the best biologists in the world to come visit and work with us. And I hope someday you will be some of those people to come visit us. Thank you. Yeah, sure. I came all this way. Why stop?
So we're doing things at a much lower level, but we had recently this observation. We wanted to examine formatting techniques with a confocal, and a few years ago, quite a number of years ago, we had a true UV laser, which allowed us to see a fine lattice mesh in the interface nucleus, which we assumed was the chromatin and then chromatin stars.
Right, exactly.
And we're doing the same thing with 405 diode laser. Didn't actually
see it.
Didn't actually do anything.
Okay.
So which of your techniques would be best for me?
Well,
sounds like something you write into a magazine. So,
well,
Dr. Eric, can you help me?
Yeah, exactly. Well, I was just meeting with all the microscopy people a while ago, so I showed this graph on the board there. So here's the graph just virtually here. So here's your axes, right? Here is blood, sweat, and tears. Here's the diffraction limit. Here's atomic resolution. Curve goes like this. So what you do is you start with your confocal and wide field. If you don't get your answer, SIM. Start with SIM, okay? If you don't get your answer with SIM, then you can start thinking about nonlinear SIM or PALM or STED or whatever. But if you could see it with UV light before, okay, you're gonna, of course, do quite a bit of damage with your UV light if you looked at it live. But anything that you could see with, at say, what you could see at 250 nanometer with UV light, you can see at 500 nanometer with SIM, okay? So that is absolutely where I would start. And if that didn't work, you might even try some of these sort of single molecule techniques like James was doing in order to show, where the hot spots of transcription were. If you had a good marker for euchromatin versus heterochromatin. I think localization microscopy, live or fixed, could possibly get there. But I would start with SIM.
I touched on the data storage issue, and that was certainly something SPIM hit very quickly, the flattening of the data that's required to store it now.
Yeah.
And mostly with even conventional microscopes, we're all running into that wall.
Yes.
Does Janelia have any particular tricks or uses to overcome this storage issue?
Well, certainly, again, for light sheet, Philip Keller, who's at Janelia and does a lot of light sheet work, has made tremendous strides in compression of the data. But a lot of his emphasis is just on, tracking of cells, right? So that can get compressed out the wazoo. There's a complexity to a lot of the lattice light sheet data that makes compression a bit harder. We use the standard compression tools when we can, but again, first we do, we always just store the process data, the deconvolved data and so forth, because it's just far easier to retake data than it is to store multiple copies of everything. But we compress, but we can't compress to the extent that you can in some cases. So better compression technologies would be great. What would be even better is ways of analyzing the data and extracting the information content you really want, and then only storing a subset of the resultant data. But one of the areas that there's many things as a microscopist I get depressed about. And one of the things is in terms of how I think it's going to be very difficult to find ways of quantifying the diversity of data that lattice light sheet gets in some generalizable way. I think there has to be probably custom solutions for a huge range of problems. And we basically need, guys like, I work with this Gautam Danuser in the States, who's one of the best quantitation guys of imaging that I know, we need to clone him 10,000 times over, okay? There just hasn't yet been even programs in the States, I don't know about here, in order to bridge that gap of you know, the information and the computer people with biologists. But it's so essential.
Any last question?
You've showed some really lovely adaptive optics images, but it seemed that those images benefited even further when you applied deconvolution. So I was wondering, why is that? Is your wavefront aberrometry so much better than your deformable mirror?
No, so this is another one of my bitches about the super resolution data that I see, is that so basically when you do adaptive optic correction, what you're doing is you're coming back to an ideal focus, all right? And you're doing that, and you're not doing that post hoc. You're doing it as you image, okay, which is essential. And so you get that, but the purpose of deconvolution is as follows. Every microscope is a filter, a spatial filter, in which low-frequency, large, squishy structures are passed with high amplitude in your image, and tiny little fine features are attenuated. So that's what's known as the optical transfer function in microscopes. So it goes like this, okay? But the thing is that therefore, when you look at a non-deconvolved image by any microscope, you are looking at a poor representation of what the object is because it has gone through this icky filter, which has amplified this, the squishy stuff, and de-amplified or suppressed the fine stuff you want to see. If, however, A, you have enough signal noise, and B, you have high confidence in the shape of that curve, you can say, oh, well, this thing, I know this was suppressed by a factor of 10, let's boost it by a factor of 10. That's what deconvolution does, when done right, okay? Now, of course, if you go to the point where the noise is greater than what's left of the amplitude of the structure here, and you amplify that, you're just amplifying noise, right? And then you get shitty artifacts and all the rest, which is something which is endemic in SIM when people don't know what they're doing in reconstructions in SIM. So my belief is that in this argument I just made about doing wide field confocal SIM, I would add in that wide field deconvolution, confocal deconvolution in that trajectory. But you need to know how to do it right. And it's not hard to know how to do it right, but again, there's still a learning process for people to know how to do it right.
I said it was the last question, but I'm going to take one more because the hand went up at the same time.
Okay, so you mentioned that you have a lot of people that come to image, and we can do the imaging now, and then they struggle for life with analysis. So I would be one of those people. What is your expertise on the analysis, and is that also something that you'd be keen to collaborate on?
Yeah, we collaborate with a few groups on a few problems, like UCO's one that I showed on the EB1, and we've got there's several groups who are doing stuff in mitosis where there's sort of a similar set of questions being asked. And so with Gowden's that I mentioned before, we're collaborating with him on a package to do some analysis, you know, like kinetic core motions and things of that sort. And so it's kind of a case-by-case basis, but we're largely hardware guys in my group. And with 3 guys, right, I mean, it's like anything we do is like a big fraction of our time. So we just try to usually put people together, right? You know, say, oh, this person can help you and like that. And so we act as kind of the binders to bring people together. So if you had data and it was similar in class to something else, we would definitely put you in touch with people with similar issues. And at least you can complain each other if nothing else.
So I said at the start that there was this sort of buzz before seminars, perhaps once every couple of years. For me, this seminar reminds me of some seminars that I heard 10 or 20 years ago, where not only was there a buzz before the seminar, but the inspiration that you're left with after the seminar can impact the research you do for many years to come. And I think that's really been true of Eric's seminar today. I really love the notion of well-funded small groups that collaborate each other. I think your seminar has indicated what can be shown by a really respectful and honest review of history as well. I love the fact that you referenced the people that you worked with so generously. I think you've shown us that science can be fun, and for the students in the room, you've extended the number of acceptable expletives that can be given. I really thank you all. Thank you for making the trip. Thank you.